Yeah, okay. Yesterday or last time, we're talking about the communication tool. The main focus is about the POC study, proof of concept. What I would like to achieve with the lecture, both yesterday and today, is to be sure that you can understand the proof of concept study is very, very, very difficult. POC, although it sounds like simple, it's just two treatment groups. The test drug group against the placebo group, and the test drug group is the highest possible dose, which is MTD, maximum tolerable dose, but sometimes a dose sort of slightly lower than the MTD. So POC is that you use this study to make a go, no-go decision. In other words, after the concept is proven, you should be able to say, okay, we're going to make a clear go decision, or we're going to make a clear no-go decision. And the difficulty is that either decision, there could be a mistake, could be an error. In other words, if the drug is truly a placebo, it doesn't work, but purely by chance, it looks like it worked, then you made a go decision, and there will be a lot more investment of this to develop this drug, and those investments could be wasted. So it's a type 1 error, false positive. On the other hand, if you make a no-go decision, but it turned out this drug really worked, and then you gave up a potentially very good drug. So it is a very difficult decision, a very difficult problem. And then we have to design a proof of concept study with limited sample size. And let's try to see what I mean. All right, we look at this graph. Here is a placebo distribution. Remember, I'm assuming positive means better. All right, so if delta is on the positive side, we say the drug works. In other words, if delta is on the negative side, the drug is worse than placebo. We're not interested in developing that drug. That actually would be a very easy decision, right? If your test drug is worse than placebo, you're going to stop developing it. Now, here's a placebo distribution. And here's a test drug distribution, all right? So this is your null hypothesis. Under null, your test drug is just another placebo. In other words, you cannot differentiate the test drug from placebo. That's your null distribution. And this is your alternative hypothesis or alternative distribution. If the drug works, that means the drug could deliver a delta unit of treatment benefit, right? Now, yesterday I mentioned a few things. One being that given the limited sample size, we have to increase delta, we have to increase alpha and beta to make the study feasible. So keeping in mind that this delta has been somewhat inflated, all right? And we're trying to calculate the sample size based on the given alpha and the beta. Now, like I said, if I standardize the delta by divided by its standard deviation, here we're talking about the standardized delta. So this difference really reflects the distance between here and here, which is Z alpha, right? From here to here. That means if you observe something to the right of this bar, you would reject the null hypothesis, okay? So from here to here will be Z alpha. I think that's easy to understand, all right? Now, what about beta? Right, beta is a false negative. That means when the drug really works, that means under alternative. But you your decision is that the drug does not work. So what would trigger you to make that decision is because you observe something to the left of this bar, right? So in other words, from this bar, you're looking at this alternative distribution. This side is beta. This part of the probability is beta. So what is Z beta? Z beta is from here to here. As a matter of fact, I should say absolute value of Z alpha and absolute value of Z beta. Okay? So the distance from here to here is Z beta. So from here to the left is beta, is a probability of false negative. And of course, from here to the right, this huge area is power, right? Power equals 1 minus beta. So that's how we understand these two curves. Now, how do we calculate sample size? So when we calculate sample size, if I have a very small sample size, then what? These two distributions are kind of like this, right? Very flat, two flat distributions. The null is flat, the alternative is flat. That's when you have small sample size. So how did you calculate sample size? You calculate sample size such that the curve will increase, right? Because, I mean, the peak will increase and the spread is getting narrower and narrower when you increase sample size. So you keep on increasing sample size. So when you increase sample size, your distribution becomes sharper and sharper, right? When you increase sample size, you actually, this point is increasing up to here. And this point is increasing up to here. Up to what time do I stop? At what sample size do I stop? When my sample size arrived at this point, such that from here to here is Z alpha, from here to here is Z beta, then I stop. And that'll be my sample size. So that's how you calculate sample size. In other words, you calculate sample size, you look at the sample size formula, you're basically trying to set up this value being this value at the squared level. All right, when you square both sides, and then the sample size will give you that this delta squared equals Z alpha plus Z beta squared. That'll give you the sample size. Of course, you have to adjust for standard deviation, the other things. So that's how we calculate sample size. All right. Now, it is absolutely critically important to understand these two curves, to understand what this represents. This is a critical point. All right. Now, remember, under null, that means the test drug doesn't work. All right. The test drug truly does not work. Now, when you observe a small p-value, So how small? This probability is less than alpha. Again, let me reemphasize, we're only interested in one-sided, okay? In clinical drug development, the only two situations you're considering two-sided p-value or two-tail probabilities is when you're doing bioequivalence or biosimilarity. Other than those situations, you're always interested in one-sided. So this is one-sided alpha, or this is one-sided alpha. Right? If you observe a p-value over here, I mean, you observe data. I mean, after the trial is completed, you observe the data point here, and you would have a small p-value, and you claim, all right, p less than alpha, right? P less than alpha, because this area is alpha. From here to the right, that's alpha. You observe p less than alpha, and you claim the drug work. You claim statistical significance. But in fact, this is under null, right? When the truth is that the drug does not work, and you claim the drug works, you're doing what? You're making a type 1 error. You're rejecting a true null hypothesis. A true null hypothesis being the test drug does not work. It's just another placebo, all right? And then you're making a mistake. But of course, if under null and you accept the null hypothesis. All right, you observe a p-value over here. You accept the null hypothesis. Under null, you're making the right decision. Okay, not a problem. Now, what if the test drug really works? It's under the alternative hypothesis. So this is when the test drug really works. And then you observe a p-value from here less than alpha. You reject the null. You're making the right decision because the drug really works and you reject it. You're fine. Now, what if you observe something on this side, but the drug really works under alternative, and then you accept it null? Then what happened? You're making a type 2 error. It's false negative because the drug really works, but the data tells you the drug does not work. You accept the null, but in fact, the alternative is true. All right. The fact is, for a POC study, proof of concept, you had to make a go, no-go decision. So after the study completes, you have to make a go, no-go decision. There's nothing in between. You cannot say, well, I cannot decide. Well, if you cannot decide, what happened? You're delaying the drug development process. The amount of time it takes you to decide actually will cost your patent life. Okay. So for one more day of delaying this decision, you're sort of losing 1 day of your profit after the drug is marketed. All right, so time is very tight. And so you want to make a decision. All right, you don't want to be, well, I cannot decide. If you cannot decide, that is what? That is a risk of inconclusiveness. And we will talk about risk of inconclusiveness. So you have to make a decision. The decision will be a go decision or no-go decision, depending on your data. Because if p-value is less than alpha, you make a go decision. P-value greater than alpha, you make a no-go decision. But either decision, you're going to make a mistake. Now, keeping in mind that you can only make one kind of mistake. You have to make one decision. If you make a go decision, the mistake could be a type 1 error. If you make a no-go decision, the mistake could be a type 2 error. Only one of the two. All right. Now, I want to talk about this as a communication tool, mainly because this discussion should happen after the study is designed, after the trial is ongoing, but before the blind is broken. Okay. Now, so we did this. That's how we designed the study. We calculated the sample size based on all these assumptions. All right. Assumptions of delta, assumptions of alpha, and beta. Based on all of this, of course, sigma, we've already considered sigma in part of the delta. So you consider all these quantities, you came up with a sample size, you make the study doable. Right? Given the limited investment, given the ethical considerations, your sample size is restricted, fine, for that given sample size. You come up with a framework of alpha, beta, delta, sigma, you come up with a sample size. Now the study is ongoing. So while the study is ongoing, and before you're breaking blind, now is the time for the statisticians to sit down with the team and discuss the consequences, okay? So here's what I do. I go to the team, I sit down, and I lay out this picture, all right? I explain to them, I said, this being placebo response, and this is a test drug response, all right? The test drug should be superior to placebo by delta amount, all right? And of course, I will have to translate this back into the raw delta level rather than delta over sigma. But anyhow, I got these numbers on hand, and I sat down and discussed with them. I said, now listen, if you observe a p-value so tiny, it's from here to the right. All right, in other words, your drug effect is greater than delta. And that's an easy decision, right? Everybody look at that data to say, well, the go decision, all right? People are very happy. We have a very good drug, you know, from here on to the right. It's a clear go decision. Well, there's not much argument at this point in time. All right. Now, what if you observe something here? It's even worse than placebo. The test drug is even worse than placebo. Yeah, people will be disappointed, but it's an easy decision. It's a no-go decision. There's a clear no-go decision. That's not a problem. Okay, so if I observe something from here to the right, it's a clear go decision. It's clinically meaningful. I mean, it's greater than delta, and it's statistically significant. The p-value less than alpha. That's a clear go decision. And from here to the right, to the left, is a clear no-go decision, and that's easy. The question happened: what happened if you observe something in between? That's where most of the cases happens. I mean, drug development. The most of the drug development program, you observe something between here and here. Very rarely you see something on this side. I mean, when that happens, everybody agrees it's a good drug. All right. So if the result, after you complete the study, you found the results right here, should you make a go decision or no go decision? All right. Now, what does it mean by over here? By over here, that means you're statistically significant, but clinically not meaningful. Right? Should you make a go decision or no go decision? All right. Now let's look at the consequences of a go decision. Consequences of go decision is that you're controlling your type 1 error rate under alpha. You make a go decision, that's fine. All right. Regardless, clinically meaningful or not, your alpha is protected. You can make a go decision. Okay. Now, what's the consequence if you make a no-go decision here? If you make a no-go decision here, your type 2 error is greater than beta. Remember, this part to the left is beta. If you make a no-go decision here, your type 2 error is inflated to be 0.5, right? So any points between here and here make a no-go decision is really to the left of this point. That's 50% beta type 2 error rate. All right? So depending on the drug, depending on the team, people may discuss different situations, different scenarios. They can make a go or no-go decision. All right. The most difficult part happens, the data is right here. And for those of you who have experiences developing drugs, especially in POC stage, most cases you're going to see something here. Okay? Something in this area. And if you're a statistician, you say, well, that's not statistically significant, it's no go. All right. But when you talk to the to the team, when we talk to the physicians, clinicians, the clinical partners, you show them this value. They say, 90, this is a wonderful drug. It's a clear go decision. I say, wait a minute. This is less than delta. We use this delta to power the study. That's our assumed treatment benefit. And then they'll say, 90, remember, when you try to calculate sample size, we have to increase the delta so that you can make the sample size feasible. So that delta has been inflated. They say, if you observe something here, it's going to be a wonderful drug. It's definitely a go decision. Okay, I say, fine, we can accept that, right? And I'll say, what if you observe something here? And oftentimes they'll say, well, that's not a drug. I mean, it's not much different from placebo. So you keep moving this point. If you keep moving this point between these two extremes, they're going to settle on a particular point. They'll say, okay, 90, below this point is no go. I say, fine, let's have this discussion. And we agree below this point is a no-go decision. And I will document this point. to say this is a reference value. All right, I call it lower reference value, or LRV. This is lower reference value. And I call this point, delta, as a target value. All right, TV, target value. LRV, lower reference value. All right. So we can agree that if what you observed is below LRV, it's a clear no-go decision. And if you observe something above delta, that's a clear go decision. So with the discussion, we can come up with this agreement, and usually I document it. All right. And now we leave this area as a gray zone. All right. A gray zone is what? A gray zone is an area of inconclusiveness. Okay. So what I just said was this. All right. Statistical hypothesis testing, your null is the test drug is worse than placebo or equal or no better than placebo, or your alternative hypothesis better than placebo, superior to placebo. All right. One-sided test. All right. That's a one-sided type 1 error. The distance between Z alpha and delta, reflects the absolute value of Z beta. All right, this area. All right, delta equal to Z alpha plus Z beta. Okay, if delta hat is greater than delta, all right, your observed value is greater than the target value, then a clear go decision. If your delta hat is between Z alpha and delta, then you still make a go decision. Your type 1 error is reserved, is protected. Now, here's a problem, right? We talked about in this area, if you make no go decision, in other words, in this area, you make a no go decision, then type 2 error is inflated. Now, if it's less than alpha, the no go decision is made. There's no inflation of type 2 error. Okay. Now, if delta hat is less than Z alpha, but the team inclined to make a go decision, knowing that type 1 error is inflated, and this is a case where a clear communication of the risks are necessary. So these are the discussion we have to have in communicating the risks the team is willing to take. All right. I will get to the risk of inconclusiveness later. All right, that is a gray zone area. But if we can come up with POC, if the concept is proven, then we can move into the dose ranging study. Okay. Now, dose ranging study. So let's go back and think about the objective phase 2. All right. Well, then let me go back to objective phase 1. Phase 1 is supposed to deliver PK and MTD, right? You need PK and MTD to help you design phase 2 studies. Now, the two deliverables of phase 2 are POC and dose ranging. All right. In other words, if POC, from the POC study, you come up with a no-go decision, then you're done. All right. You're not going to further develop this drug because you already made a no-go decision. But if you make a go decision, then the next objective is achieve dose ranging. All right, because you need to come up with a lower limit of the dose range from phase 2, and then you can select one or few doses to move to phase 3. So dose ranging is absolutely critical that you have to achieve in phase 2. All right. The reason we had to establish the lower limit of the dose range in phase 2 was because in phase 1 you didn't get to see efficacy, right? Mean effect dose is based on efficacy. You have to do it in patients with a disease. So in phase 2, it is critical to establish this lower limit of dose range, which which is minimum effective dose. All right. Now, typical dose ranging studies, let me give you an example, would be placebo, low dose, medium dose, high dose. You have 4 treatment groups, okay? Placebo, low, medium, high. And then you look at each dose compared to placebo. Do you have statistical differences or whatever? And then you select a dose. You select a few doses to move on to phase 3. And also from the dose ranging study, you can establish some lower limit of the dose range to see if the low dose is too low or medium dose is not high enough or things like that. All right. So dose ranging study is typically a parallel dose groups, placebo controlled, the original treatment limited by animal tox coverage, and many doses of test drug. So that's a typical dose ranging trial. Objective is to explore a range of efficacious doses. All right. So we look at minimum effect dose, mean ED. All right. Imagine the difficulty in a POC study. All right. So I spent some time in the previous lecture and the most part of the first lecture to impress you that how difficult it is to make a POC decision. All right. So POC is very, very, very difficult. All right. We can design the study, and with limited sample size, with our assumptions and with the kind of risk we're willing to take, we came up with this design, and we started conduct the study. And after the study results came out, we started to make a go, no-go decision. And we know it's very, very difficult. All right. So it's very difficult. But that decision was based on MTD. All right. From dose ranging design, there are multiple test doses. All right. So you're looking at minimum effect dose. You're looking at multiple doses. When each dose is compared to placebo, there's a POC decision. All right. In other words, you're looking at each dose compared to placebo. It's just like you're doing POC, right? Is this dose good enough? Is this dose good enough? Is this dose good enough? And we have to determine which dose is efficacious. And after that determination of each dose, you're going to select the lowest one. So minimum effect dose is mission impossible. That is why for most of the drugs approved and available on the market, we don't know their minimum effect dose. Right? Minimum effect dose is very, very difficult to get. It's mission impossible. POC is already very, very, very difficult. Mean ED is kind of impossible. Right? But 19, you said the objective phase 2 is to come up with a lower limit of the dose range, which is minimum effective dose. But now you say it's mission impossible. So how do you get to that point? All right. So here is the ideal. All right. You would hope to come up with some understanding of minimum effect dose. But in practice, it's very difficult to determine that dose. Why? I mean, statistical significance is not easy to achieve. All right, because you have sample size, your sample size are limited. And furthermore, if you have multiple doses in one trial, you can deal with multiple comparison adjustment. All right, once you deal with multiple comparison adjustment, your sample size will increase to be a very large number, and it's definitely not feasible. But we have to come up with some idea of where the minimum effect dose is about, because if you don't, I give you the example of this other company. They sort of marketed 100 milligram at other countries, and then when they came to US, they had to do a dose ranging trial, and they had to bring down the the market dose from 100 milligram to 25 milligram. It's very expensive. So let's first think, what does it mean by dose range? All right, suppose I have two studies. Study A is designed with placebo 20, 40, 80 milligram. Study B is designed with placebo 0.11 and 10. So which design has a wider range? All right. Is A have a wider range or B have a wider range? All right. So I should first define a dose range for a given study design. All right, here it is. The dose range for a given study is defined as a high dose divided by low dose in the design. All right, remember it's division. High dose divided by low dose. It's not subtraction. Right, if you look at subtraction, 80 minus 20, you got 60. 10 minus 0.1, you got 9.9. Looks like this one is wider. No, that's from subtraction. But by division, 80 divided by 20 is 4. 10 divided by 0.1 is 100. So B has a much wider range. Design A has a range of 4. Design B has a range of 100. And I'll come back to you as to why we use division instead of subtraction. If we get into dose ranging studies, then how do we design that? First question will be number of doses to be tested. All right. So how many doses we're going to test? Remember the case I told you about placebo, low, medium, high. You have 3 test doses against placebo. All right. Do you need active control? And the true challenge is your dose spacing. All right. What does it mean by dose spacing? Dose spacing means the difference between the high dose and medium dose, between medium dose and low dose, and between low dose and placebo. These are called dose spacing. Of course, the choice of endpoints, the length of study, these are all necessary considerations in designing a dose ranging trial. Now, why do we separate POC and dose ranging? All right. As many of you in the industry now, in the pharmaceutical industry now, you understand that in most drug development programs, they combine these two together. So in other words, their first phase II study is a dose ranging trial. When your first phase II is a dose ranging trial, that is a combination of POC and dose ranging into one trial. But traditionally, we didn't do that. Traditionally, in the past, we separate them. Right. So why do we separate? Because we're not sure if that drug works. All right. So you go through a POC. If the concept is not proven, then you don't do it. All right. So we're not sure. And there are formulation limitations. Okay. I will come back to the formulation issue later in designing dose ranging trials. Now, suppose you have already formulated 5 milligram tablets. All right, so we have 5 milligram tablets. All right, that's good. You can test them 5, 10, 15, 20, up to very high doses. All right. But there's no way you can test for 3 milligram or 7 milligram. All right, so there are formulation limitations. And from phase I, going to phase II, you're going to extrapolate. Phase I, you can only observe PD markers, pharmacodynamic endpoints. Now, phase II, you're looking at clinical efficacy endpoint. You're working with patients, right? So you're collecting patient efficacy data. All right. Now, of course, the investment, the cost will be prohibitive, because if you don't have enough investment for dose ranging trial, you want to start with a POC, so you separate them. And of course, there are possible ethical concerns, because you don't know whether the drug works or not. I mean, you expose a lot of patients to it, there might be ethical concerns. But the POC decision is very, very important, very, very critical. All right. Now, the first phase II study has to be a proof of concept study, because before proof of concept, you don't have to do a lot of formulation and develop different kind of pills or capsules or stuff like that. All right, because the concept is not proven. You don't know whether the drug works or not. All right. And also, before POC, you don't have to order large quantity of raw materials. Right? This is very important from a development planning way of thinking. All right. So in the previous lectures, we talked about CDP, clinical development plan. From the planning stage, all right, you would say at a certain time, the drug will move into phase 3. All right. If the drug goes to phase 3, you need to prepare a large quantity of test drugs. We call it clinical supply. You have to have sufficient test drugs for your phase 3 studies, right? Because phase 3 are long-term, large-scale trials. You may recruit hundreds of thousands of patients, and you have to follow them for years. So you have to have enough drug supply. Now, if you have enough drug supply, if you want to have all the, you want to have sufficient drug supply when you engage in phase 3, then you have to order the raw materials to produce these large amount of phase 3 drugs way ahead of time. Okay, but before POC, you don't have to do that because the drug may never get to move beyond phase 2. So you don't need to do that. Or if you do that, it's a waste of effort, waste of money. But once the concept is proven, that means the company is determined, the project team will move forward into phase 3 development, then you better have sufficient drug supply, sufficient clinical supply there. And then we need to order large amount of raw materials. Now the formulation, again, is a very critical part because phase 1, in phase 1, you may not have all the solid formulation yet. All right? In phase 1 trials, you can do powder, you can do liquid. I mean, because phase 1 units are mostly, we call it inpatient units. I mean, even though they're healthy volunteers, they come to the phase 1 unit, they stay in the unit for like 24, 48, 72 hours. You'll follow the PK and all that kind of stuff. At that point in time, you can do flexible dosing, or you can have powder or liquid because the study nurse will give you the right amount of drug. But phase 2 and phase 3 are what we call outpatient studies. So either lower blood pressure, treating rheumatoid arthritis, or whatever disease or condition. Patients, they're going to live their normal life. Only at the clinical visit, they come to the clinic. Or if it's not clinical visits, they're at home, they're doing their businesses. So you cannot give them powder form or liquid form to let them measure the amount of drugs they take and things like that. So you have to have solid formulation. All right. So the formulation has to be ready before the phase 2 is started. All right. So during the phase 2, you're still sort of improving your drug formulation. All right. And then if the concept is not proven, you're not going to order a large quantity of raw material. And also, to engage in long-term trials, phase 3 trials could be half year, 1 year, or longer. You have to have long-term toxicity studies. Long-term toxicity studies, you probably have to follow some larger animals like dogs or monkeys or cows for a long time. 13 weeks, 26 weeks, 52 weeks. All right, if the concept is not proven, you don't have to start your long-term study, long-term tox study. But once the concept is proven, then you have to start the long-term studies, long-term tox studies. All of these are costly. So before POC, you don't really have to engage in a lot of investment in these activities. But once concept is proven, you have to do all of these. So a critical no-go decision is very critical. And we need to avoid inconclusiveness. So what is inconclusiveness? The clinical trial process goes like this. Design, conduct, unblind, you got results, and you make a decision. All right. The question of POC is to go or not to go. So we had to make that decision. How do you make that decision? And also this decision has to be made. In other words, there's nothing in between. Delay this decision will impact formulation, order of raw materials, and toxic studies. Inconclusiveness happens between study results and decision. Between study results and decision. Okay. Now, like I said earlier, if you underestimate MTD, what will happen is that you did, well, if the true MTD is 200 milligram, but your phase 1 told you it's 100 milligram, you did a POC was 100 milligram against placebo, and the results look good, but it's not good enough, and the drug is very safe, you would consider whether you have to do another POC. That amount of time is inconclusiveness. All right. So between your readout of your first POC result and you finally decide to make a go or no go decision, that amount of time being the risk of inconclusiveness. All right. If you go to most of the drug companies, what happened is that there's a project team that developed the drug, and if the clinical trial is successful, then everybody celebrates, says the team did a great job. You know, the drug is successful. Now, if the drug is failed, then the team member seems to be disappointed. It looks like they're doing something wrong. But whether the drug is successful or fail has nothing to do with the team. Right? So the evaluation of a team should be separate from the evaluation of the drug. Drug can be good or bad, it's the drug itself. Right? A good team is what? A good team could help make a clear decision. All right. So what would be a failed team? A failed team is that they cannot make a clear go, no-go decision. In other words, there's too much inconclusiveness. So if we go back to that gray zone problem between LRV and TV, lower reference value and target value, right? You want to minimize that region, all right? Because if that region is too big, it's going to be more likely to lead into inconclusiveness. So after results are ready, there's very little a statistician can do, all right? Remember, we look at probability before breaking blind, all right? In my earlier career, I remember every time the study's unblinded, I'm going to be very busy. We're going to analyze data from left to right, all right? All kinds of subset analysis and different kind of models, different things. Sensitivity analysis, exploratory analysis is a lot of work, all right? So in other words, as a clinical statistician, after blind is broken, I became a pair of hands. I'm just busy from left to right. But where the value of a clinical statistician happens before breaking blind, like those POC communication, that's critical. All right, that's where statistics could provide value for drug development. All right, so after study results are ready, there's very little I can do. I can only perform a lot of analysis. The data is the data. The data are out there. All right. But the key contribution of clinical statistician is before you see the data. Is how do you design the study? How do you understand the risks? Okay. So the critical time for statistician to help the team is at the design stage. Clearly communicate type 1, type 2 risks. Right? That's why the communication tool is absolutely important and define go, no-go criteria. Right? That's where statisticians make real contributions to the company. So we talk about dose ranging, all right? When people think about dose ranging, when people think about dose response, that's a natural curve that came in mind, okay? So as your dose increases, your efficacy increases, right? Well, the curve looks nice, right? But it's only a theoretical curve, all right? Now, if I keep increasing the dose, the response does not increase. Then what? You say the drug doesn't work, right? If it stops here, then you say, well, that's the best efficacy the drug can ever deliver, right? You keep on increasing the dose, the response does not increase much. Okay. So in other words, if the drug works, if you keep increasing the dose, at certain places, the efficacy response should increase. All right. At certain point, the efficacy should be increased. All right. Of course, in some drugs, you keep increasing the dose, the response or the efficacy could decrease. It actually happens. All right. That's what we say is non-monotonic. All right. So this is a monotonic dose response curve. All right. So when dose increases, efficacy increases. That's monotonic relationship. But if your dose keep increasing, it decreases, then the curve, the dose response curve is non-monotonic. All right. In my experience, the non-monotonic dose response curves happen in CNS trials, CNS being central nervous system drugs. All right, like drugs for depression, for anxiety, for schizophrenia. For CNS drugs, they tend to not have a monotonic dose response curve. But in most cases, we can assume monotonicity. All right, so when dose increases, the response should increase. If you keep increasing the dose, the response does not increase. That means you don't have a drug. Now, if this is the efficacious drug, we increase the dose at some point of the doses, the efficacy should increase. Okay. Now, if I'm looking at this curve and I'm designing my phase 2 study, dose ranging study, all right, and I say if the dose response follows this curve, in IT you have to design 3 doses plus placebo. Here's how I'm going to allocate my 3 doses. I'm going to allocate the dose here, right here. I'm going to allocate the dose here, right? I'm going to allocate the dose here, right? So I'm going to allocate 3 doses here. That's okay. That's good. All right. Now the difficulty is that this increasing part of the dose response curve, that's the most critical part of the dose response curve. In other words, however many study doses you want to allocate, you want to capture this part of the curve, right? Now, suppose I'm very lucky. I got a very good drug that my maximum tolerable dose is 1,000 milligram here. 1 gram, 1,000 milligram. I got a very wide range of doses I can test. All right. Now, this curve where the efficacy could happen between 0.0001 milligram and 0.001 milligram. All right. This curve could happen between 0.0001 and 0.001. Or it could happen between 0.001 milligram and 0.01 milligram. Or it could happen between 0.01 and 0.1. Or it could be between 0.1 and 1. Or between 1 and 10. Or between 10 and 100 or 100 and 1,000. That I don't know. All I know is concept is proven. I did 1 gram against placebo. Concept is proven. The drug works. Pretty safe. Now, I'm designing a dose ranging trial. And I'm allowed 3 doses. All right, so I got 0 being placebo here, 1,000 milligram here. So the 3 doses, how I'm going to allocate. All right, now if I know is between 100 and 300, I'll probably allocate like 100, 200, 300, or something like that. but I don't know. Right? There's infinite possible curves, and I don't know which curve that will be. And I'm still assuming monotonicity. I'm assuming the efficacy will not decrease at higher doses, right? Given the assumption of monotonicity. And there's infinite possible of dose response curves. And how am I going to allocate my 3 doses for the phase 2 testing? All right. Now, this curve is only helpful when we think about dose response relationship from a concept point of view. So it's only a theoretical curve. In practice, you never see this curve because in practice, all you see are just a few discrete points. In other words, at different doses, you're going to allocate a certain number of patients. You're going to measure their responses. And then you're going to see some scattered data points. You try to use those scattered data points, guess what the curve would be. Okay, so in reality, you never see this curve. The dose response also changes between individuals, right? For example, this might be your dose response for that drug. This may be my dose response for that drug. This could be dose response of another patient. So we don't get to see individual dose response. The best we can see is some kind of average response like this curve, all right? Because in clinical trials, we're looking at population phenomena. We're not looking at the individual phenomena. So we have a group of patients. We use a group of patients to try to guess where the response is about. All right? Now, the dose response have the efficacy response and the toxicity response. Okay? So we see as efficacy increases, as dose increases, efficacy increases. But also the toxicity increases. As dose increases, toxicity increases. Okay? Now, suppose we say this is 100% response, either 100% efficacy or 100% toxicity. This is 100%. And this is 0%, which is placebo. Placebo says here, all right? Now you look at 50% of the response, right? You look at the 50% response. And this horizontal line, if that intersect at the efficacy curve, that dose, we call it ED50. The dose that delivers 50% of efficacy, all right? Capital E, capital D, and 50 being subscript, all right? ED50. So in other words, you can do ED20 here, right? That's ED20. Or ED80. Oh, ED80 is here, right? ED80, ED20, all right? The dose that delivers a certain percentage of efficacy. Now, when that curve intersects with toxicity, that dose we call it TD50, the dose that delivers 50% of toxicity, all right? And people tend to say between ED50 and TD50 will be the therapeutic window or the therapeutic index, all right? So they say this range of doses being the therapeutic window or therapeutic index, okay? Now, what I would say, well, minimum effect dose, mean ED, could be ED40, could be ED60, depending on how you look at that drug and for the efficacy and the response and your measurement scale and all that kind of stuff. But I think TD20 is already toxic, so I think I would stop here. So I would say the dose range is between here and here, you know? That's my point, that's my personal point of view. So what would be an ideal drug? Right? An ideal drug would be like this. The efficacy curve starts at very low. The very low, it shoots to 100%. And the toxicity curve is flat. You keep on increasing the doses, it's still safe, it's not toxic. Okay, so that's an ideal drug. But in reality, in practice, most drugs, they only have a reasonable therapeutic window, therapeutic index. And some drugs has very narrow therapeutic window. The efficacy curve and the toxicity curve very close to each other. All right. Now, if you're looking at chemotherapy, the efficacy curve and toxicity curve all almost get together. All right, because for chemotherapy, you're trying to kill the tumor cells. So we call these drugs cytotoxic drugs. So they may be killing your normal cells also. All right. Well, in practice, the best dose-response relationship you could get is probably look like this. All right, you got many study doses, and at each dose you have a few subjects. You measure their efficacy response, and you collect the data. Okay. So I will stop here for this lecture, and I will continue into our next lecture next time. Thank you.